Seeds are remarkable reproductive units of flowering plants, angiosperms and gymnosperms, acting as powerhouses of plant reproduction. They contain all the necessary components for a new plant to begin its life. Seed morphology and structure. While seeds vary greatly in size, shape, and appearance, their basic structure generally includes seed coat, testa. This is the outermost protective layer of the seed derived from the integuments of the ovule. It safeguards the delicate internal parts from mechanical damage, desiccation, drying out, pathogens, bacteria, fungi, and harsh environmental conditions. Testa, the outer, often hard and thick layer providing structural support and defense. Tegmen, a thinner inner layer, usually membranous, which helps regulate water and gas exchange. Hilum, a distinct scar on the seed coat, marking the point where the seed was attached to the ovary wall or fruit via the funiculus, stalk. It's like the navel of the seed. Micropyle, a tiny pore or opening, typically located near the hilum. This opening allows for the entry of water and oxygen, which are crucial for germination, and in some cases, the entry of the pollen tube during fertilization. Embryo. This is the miniature, undeveloped plant inside the seed, formed from the fertilized egg, zygote. It remains in a dormant state until favorable conditions for germination arise. The embryo consists of embryonal axis, the central axis of the embryo. Radicle, the embryonic root, which is the first part to emerge during germination, anchoring the seedling and absorbing water and nutrients. Plumule, the embryonic shoot, which will develop into the stem and leaves of the new plant. It consists of a short stem with tiny leaves and a growing point. Cotyledons, seed leaves. These are embryonic leaves that play a vital role in nourishing the developing embryo. Their structure and function vary depending on the seed type. Epicotyl, the region of the embryonal axis above the point of attachment of the cotyledons, giving rise to the shoot system. Hypocotyl, the region of the embryonal axis below the point of attachment of the cotyledons and above the radicle. It elongates during germination to push the plumule above the soil surface. Endosperm. This is a nutritive tissue that surrounds or is adjacent to the embryo, providing food reserves for its development and during early germination. It is typically triploid, 3N, in angiosperms. The endosperm stores nutrients primarily in the form of starch, proteins, and lipids. In some seeds the endosperm persists, while in others its nutrients are absorbed by the cotyledons during seed development. Aleurone layer. In some seeds, like cereals, this is a protein-rich outer layer of the endosperm that plays a role in enzyme secretion during germination. Seed composition. The primary components of a seed's composition are carbohydrates, stored primarily as starch in the endosperm or cotyledons, providing the main energy source for the developing embryo. Proteins, essential for structural components and enzymatic activities. They are stored in both the endosperm and cotyledons. Lipids, fats, oils. High energy reserves, particularly in oily seeds, for example sunflower, castor bean. Water. Though mature seeds are often in a relatively dry state, they contain a small percentage of water, which increases significantly during imbibition, water absorption, to activate metabolic processes for germination. Minerals and vitamins, present in smaller quantities but crucial for healthy seedling development. Hormones, such as abscisic acid, ABA, for dormancy and gibberellins, GAS, for promoting germination, regulate seed development and germination processes. Types of seeds. Seeds can be classified based on various criteria, primarily their structure and the presence/absence of endosperm. 1. Based on the number of cotyledons, monocotyledonous seeds, monocots, possess a single cotyledon, often have a thin, membranous seed coat fused with the fruit wall, as in cereals like maize, wheat, rice, where the grain is actually a fruit called a caryopsis. The endosperm is usually large and serves as the primary food storage tissue, for example, maize, rice, wheat. The single cotyledon, scutellum, in grasses is shield-shaped and functions in absorbing food from the endosperm. Examples: maize, rice, wheat, onion, lilies, orchids. Dicotyledonous seeds, dicots, possess two cotyledons. The seed coat can be thick and distinct. Cotyledons are often fleshy and store food, or they can be thin and membranous if the endosperm is present. Examples: pea, bean, gram, mustard, mango, sunflower. 2. Based on the presence of endosperm. Endospermic seeds, albuminous seeds. These seeds retain a significant amount of endosperm at maturity, which provides nourishment to the embryo during early development. The cotyledons in these seeds are usually thin and membranous. Examples: castor, maize, wheat, rice, coconut, poppy. Non-endospermic seeds, exalbuminous seeds. These seeds lack a significant endosperm at maturity because the developing embryo has absorbed all the nutrients from the endosperm during seed development. Food reserves are primarily stored within the thick, fleshy cotyledons. Examples: pea, bean, gram, groundnut, sunflower. Other classifications: orthodox seeds can be dried to low moisture content and stored at low temperatures for long periods. For example, most cereals, legumes. Recalcitrant seeds cannot tolerate desiccation and low temperatures and must be kept moist. For example, mango, coffee, oak. True seeds, formed from fertilized ovules through sexual reproduction, containing genetic material from both parents. Apomictic seeds, formed without fertilization, producing offspring genetically identical to the parent plant.